Thank you so much for the invitation to give this talk, and I'm really excited to share my lab's work. As Dr. Brumback mentioned, I am an assistant professor at UC Irvine right now. I'm a physician scientist, and my lab is a basic neuroscience lab that is very interested in understanding the neurobiological mechanisms of early life physical activity and using preclinical models to do that and learning from this information in order to then extrapolate mechanisms and interventional strategies for our children with neurological conditions or who otherwise suffer from cognitive dysfunction. So I titled my talk, The Neurobiology of Early Life Physical Activity and Childhood Neurological Health. And so hopefully some of what I share and what we're learning in the lab can be informative for how you think about patients that you see that may have cognitive impairments. Okay, so why study early life physical activity? Well, we do know that in general, as a human population, childhood physical activity is declining. And if you think about this from an evolutionary neuroscience perspective, back in the days, thinking way back when we were hunter-gatherer nations, people had to travel long distances in order to find sources of food and water and stay away from danger. And they had to use their long-term memory and spatial navigational skills in order to do this. So physical activity was baked into daily lifestyle as well as memory mechanisms and navigational mechanisms in the brain. If we flash forward to industrialization of our societies and our nations, but prior to real, advances in technology over the past couple decades, you would still see children playing outside quite a bit more often. I remember when I was growing up, we were playing outside until the street lights came on. That was a big deal. But nowadays, we're seeing lots of our kids in front of screens. And, to no one's fault, but this is just how we've evolved and how, our current society is. But this has significantly impacted the amount of physical activity that children are seeing. In addition to this, and the big elephant in the room is the impact of COVID-19 on the lifestyle of our children and their access to physical activity. And so what impact does this have on brain development and brain function? And so the impact of physical activity during childhood is also understudied. And that's another reason why we decided to focus on this area of our research. And there is a guideline that's actually been published by the Department of Health and Human Services here in the US. And it's a physical activity guideline for Americans that's very specific with regard to the amounts and types of physical activities that should be implemented in adulthood for optimizing, physical health and mental health. However, because of the paucity of research of childhood physical activity and brain health, as well as long-term physical outcomes, there just isn't much. So there are no exact guidelines that actually exist for optimal duration and intensity of physical activity in youth younger than 6, given the paucity of literature on this. But yet this is during the time that exercise may actually have the most benefit. And my subsequent slides hope to make that case more obvious. And so preclinical and clinical data are needed to inform targeted exercise guidelines for our pediatric population. And so thinking about physical activity in childhood and neurological health, from a pediatric neurologist perspective, how can we learn from the mechanisms and the neurobiology of physical activity and the cognitive outcomes of our patients with chronic neurological disorders? We do know that a number of our patients do have comorbid cognitive dysfunction, in addition to neurodevelopmental disorders where there are cognitive impairments. We also see these things with diagnoses in epilepsy, neuromuscular disorders, as well as in neuro-oncology. We see impacts on cognitive health with some of the treatments that we give as well. But sometimes this is often ignored, and for good reason, in order to treat the primary illness. But how can we also treat and optimize the cognitive health of these kids? And so can we actually learn from exercise mechanisms and how they interact with the developmental plasticity of the brain during childhood? And can we target these mechanisms precisely to then think about how these mechanisms can be utilized for interventions. And can we target these mechanisms both pharmacologically as well as with precise physical activity prescriptions? And so, in starting this work, I began with the neurobiology of exercise in adulthood because that's where the preclinical literature is. And so, in 2020 was actually the 25th anniversary of the start of exercise neurobiology, and there was an important paper that actually was published in Nature in 1995, which discovered the neurotrophin, brain-derived neurotrophic factor, as being critical as one of the proteins that's increased in the hippocampus after physical activity, and specifically aerobic physical activity. And this was found in rodents. And this finding sparked a whole 2 decades plus of research, really interrogating how BDNF promotes plasticity and memory, and specifically those plasticity and memory mechanisms that occur in the hippocampus. So I titled this slide a brief overview, but there's so much literature actually in this realm. So there's a lot that we actually do know about what physical activity is doing to neuronal function in the hippocampus. We know that it promotes increased vascularity and perfusion. We know that there's also an attenuation of local inflammatory and oxidative stress markers, as well as mitochondrial adaptation within neurons in the adult hippocampus following physical activity. As I mentioned, there's increased expression of NMDA receptor subunits in the hippocampus following physical activity in adulthood. You also get increased transcription and translation of a number of neurotrophic factors. I mentioned BDNF. These things converge to stimulate hippocampal neurogenesis during adulthood, so birth of new neurons during adulthood after physical activity. We also know that there is a synaptogenesis, or new synapses in hippocampus after adult physical activity, again in rodents. This leads to increased dendritic complexity and spine density. This process promotes this phenomenon called long-term potentiation, which is essentially the cellular correlate of learning and memory in the hippocampus. You get strengthening of synapses, and all of these mechanisms really converge to promote enhanced hippocampal function. More recently, literature has been demonstrating systemic markers that cross the blood-brain barrier and can act locally within the hippocampus to influence its function after acute bouts of exercise. Some of these include beta-hydroxybutyrate, as well as myokines, which are the factors secreted by exercising muscle. So these number of mechanisms have been thoroughly investigated in the adult brain, but not in the developing brain. So the consideration that I bring to this research is, can we consider how these mechanisms may impact a developing nervous system, and specifically developing hippocampal circuits? So in order to start thinking about this, we have to consider the neurodevelopmental milestones of the hippocampus, and we can consider these across species in rodents and in humans. So that yellow bar at the very bottom is showing that neurogenesis is a process that occurs within hippocampus throughout the lifespan. However, neuronal migration, synapse formation, synaptic pruning, as well as circuit maturation are all processes that can occur postnatally in a developing hippocampus. So the goal that I wanted to demonstrate here in this illustration is that, is there potentially a sensitive period for targeting postnatal interventions that we know promote neuronal plasticity and synaptic strengthening in hippocampus? And can we learn from exercise targeted during these critical postnatal developmental periods to understand how it intersects with the developmental plasticity of the brain and of the hippocampus specifically? So the overarching hypothesis of the first kind of project in the lab, that early life exercise, specifically during these periods of postnatal hippocampal maturation, can have a lasting impact on hippocampal function as well as synaptic plasticity. And a second arm to this hypothesis is that epigenetic mechanisms are intimately involved in exercise impacts on hippocampal function. And so I'll begin by talking about our model of early life exercise that we use in the lab with rodents. And so we have wild type mice in this particular model that are born and raised in our animal facilities. And as soon as they are juvenile, so when they're 21 days old, they are put into running wheels. And we have 3 different exercise groups. We have a group that exercise just for 1 week from postnatal day 21 to 27. So this is their 4th postnatal week of life. This is a very sensitive period in terms of hippocampal maturation and circuit establishment. We also have an exercise period that's happening in later adolescence during postnatal days 35 to 41. And this is kind of a control for that early versus later life. And then we have a 3-week exercise period that spans the juvenile and adolescent period. And the reason why we chose 3 weeks is because in adult rodent literature looking at physical activity, 3 weeks seems to be the minimal amount of exercise that's required in order to see improvements in memory function and hippocampal plasticity. The 2 readouts that we chose for this first experiment were looking at a hippocampal memory task, a behavioral task called object location memory, in which the animals have to discriminate between an object in a familiar location versus an object in a novel location. And we also looked at synaptic plasticity in the hippocampus with long-term potentiation studies. And so the way we asked the questions here, and first before I get to the results, this data just shows that both male and female mice that were included in this study ran to similar degrees, regardless of timing of exercise during early life. And so we did not see differences across the sexes in terms of amount of exercise that took place, which was important to establish. So then we set up our behavioral experiments, this object location memory task, by testing a subthreshold memory stimulus. And so what is a subthreshold memory stimulus? So typically in this task, animals require 10 minutes of exposure to the task in order to form long-term memory of it. So we asked the question, well, if we give them less than 10 minutes, we give them 3 minutes of exposure to the task, can they still learn it? And if they can't, if we preemptively exercise them, can that actually facilitate the formation of long-term memory when exposed to this shorter subthreshold learning stimulus? So we first did the 10-minute arm, 10 minutes being kind of a positive control, because we know that all animals are supposed to learn this task when they're exposed for 10 minutes, and indeed they all do. And again, these are adolescent mice. This protocol had not been done before in adolescent mice, and so it was important for us to establish that, yes, these adolescent mice do learn this hippocampal-based memory task, and this is right after the period of hippocampal memory circuit establishment. And so hippocampal circuits are functioning here and are able to express long-term memory. When we expose animals, both sedentary and our exercise groups, to the 3-minute subthreshold learning, this is when the data became interesting. So when you look at panel F, the green bars on the left are males, pink on the right for females, and the animals that exercise specifically during that 4th postnatal week, between postnatal days 21 to 27, as well as the 3-week period, they acquired this task when exposed to the subthreshold learning stimulus, and this was true for both the male and the female groups. However, the animals that did not exercise, or the animals that exercised for 1 week during later adolescence, did not acquire this task, which told us that there was likely something specific about that early life period that allowed for the facilitation of this long-term memory formation when exposed to a subthreshold learning stimulus. In addition, we performed long-term potentiation studies. This essentially involves us inserting a stimulating electrode in the circuits that feed into area CA1 of the hippocampus. And then we insert a recording electrode in the CA1. And we ask the question, if we give a pattern stimulus, theta burst stimulation, or TBS, and this type of stimulation is supposed to pattern the type of electrical activity that occurs during hippocampal learning, can we facilitate strengthening of synapses by measuring excitatory postsynaptic potentials? We did this ex vivo in slices and did field recordings. And so indeed, In collaboration with a senior scientist, Aniko Kramar, she found that animals that exercised for 3 weeks, but not the animals that exercised for 1 week during the juvenile or adolescent periods, had an increase in LTP. So only the 3-week exercisers had an increase in this type of synaptic plasticity. However, the data became really interesting when we looked at basal synaptic function. So we asked the question, if we give increasing currents to these synapses in area CA1, are they more able to respond more potently to the currents that are received over time? And so in panel C, we looked at the excitatory postsynaptic slopes for area CA1 hippocampal synapses, and found that with increasing current, both the animals that exercised for 3 weeks and the animals that exercised for the 1 week early period, but not the 1 week later period, these first two groups actually had an increase in this excitatory postsynaptic potential slope, suggesting that they actually had stronger postsynaptic potentials when compared to the animals that exercised for 1 week during later life. So we found that this was really fascinating, that actually we changed properties of the postsynaptic physiology of these animals when they were exercised during these early life juvenile periods. And so to briefly summarize what I just told you for the first half of this talk, both male and female early life exercise mice do exercise in our model. And this was important for us to demonstrate because this type of model had not actually been done before. We also showed that exercising the animals for 3 weeks during the 4 through 6 postnatal weeks was sufficient for enabling long-term memory after the week learning stimulus. This is also true in adults, so we repeated this in our adolescent mice. However, what was really interesting was that exercise specifically during the 4th postnatal week, but not during the 6th postnatal week, was sufficient for also enabling this long-term memory after the week learning stimulus, the 3-minute learning stimulus. Another thing that I didn't highlight here, but what's important to note, is that this effect persisted 2 weeks after the exercise ended. So when these animals stopped exercising, we didn't test their memory until 2 weeks later and found this enabled memory function. We believe that the enabled memory function is supported by the changes in synaptic plasticity that we saw. LTP was significantly increased only in the mice that exercised for 3 weeks, but we found changes in the basal synaptic function, specifically in those postsynaptic properties of hippocampal CA1 neurons in mice that exercised during that 4th postnatal week. And so turning our attention to mechanisms that may support a persistent effect of early life exercise on hippocampal neuronal function, we decided to turn our attention to epigenetics, which I'm sure this audience is familiar with, but to define it briefly here for us, this is modifying gene expression without altering DNA sequence. And the types of epigenetic mechanisms that I will be thinking about and talking about in this particular presentation are DNA methylation, so methyl groups that are placed directly on DNA, as well as histone modifications. And these histone modifications can be either acetylation or methylation, and they are placed on lysine tails of the histone proteins that house the DNA within chromosomes. In general, acetylation tends to open up that DNA structure away from the proteins that house them in order for transcription factors and enzymes to come in and transcribe that DNA into translatable proteins. If you methylate these lysine tails on histones, however, in general, this tends to bring the DNA closer to the histone proteins and renders that DNA inaccessible for enzymes and transcription factors to facilitate gene transcription. So the conceptual framework that we brought to this early life exercise model is that we think that the neural epigenome really sits at the intersection of gene expression and environmental experiences, including exercise, to inform phenotype. So as we all know, you're born with your genome. There's not much you can do about that. If you have a mutation in that genome, it's going to directly inform phenotype. However, the environment, as we know, can absolutely influence how that genome is expressed. And can we influence through early life environments and experience-dependent transcription how the phenome is expressed? And specifically, in our case, can early life exercise influence how cognitive function is expressed, how memory is formed and expressed due to early life experiences such as exercise? And so we then have the hypothesis, is there an epigenetic signature of early life exercise? So going back to those mechanisms that I introduced regarding histone modifications, what we are proposing here is because our juvenile exercise animals that exercise during that fourth postnatal week have this enabled learning process that persists for 2 weeks after the exercise ends, is there an epigenetic signature of this? Are we promoting acetylation on the histones of the that are associated with the genes that support memory function and learning within hippocampus? And so we created a mouse model in our lab to get at this very question. And the model that we created, we titled the EMX1 Neutrap mouse. Now, this is a mouse that allows us to, from single neurons within the hippocampus, take out the information from the nucleus, which has the DNA and all of its epigenetic modifiers, and from the same cell, take the information from the translating ribosomes. So what proteins are about to be produced in that cell. We can couple that data, so look at the epigenetic modifiers and the proteins that are going to be produced from the same cell and ask the question, how did early life exercise influence that process? And so through developing this transgenic mouse and coming up with a few techniques by combining a few that are in the that are already in the literature, we were able to develop this mouse, and it's now actually on bioRxiv as a protocol. And I'll show you some of the preliminary data from this study. And so first we had to validate that, yes, we are indeed labeling the areas of the cells within hippocampus that we think we're labeling. And so the cytoplasm is labeled with GFP, and that's where your translating ribosomes are. So we're able to pull those ribosomes and the associated translating mRNA with those. We were also able to label in red the nuclear membrane. So then we're able to pull out the nuclei with the red signal and get the nuclear DNA that has the epigenetic information. And again, this is from the same cell. So what did we find? After early life exercise, we found a gene expression signature that is very similar in adults, but we also found some interesting and new gene expression programs that seem to be different than adult exercise, which we are beginning to explore. But we were really happy to see with this EMX1 Neutrap mouse, when we early exercise those mice during that 4th through 6th postnatal week, we found an enrichment of neuronal genes. So we were able to isolate neuronal cells like we thought we were, which was great confirmation. And we saw some of the usual players. We saw an increase in BDNF and some other immediate early genes responsible for memory mechanisms, including NR4A1. We were then able to look at the histone marks, the acetylation and the methylation that actually is associated with the genes that we saw go up or down after this early life exercise. And again, this is an approach that has not been described in the literature before, to be able to couple this histone mark data with gene expression from the same cells after early life exercise. So what did we find? We found that there were 5 specific genes within hippocampus that were highly expressed after this early life exercise, not implicated in exercise neurobiology in the past, that had new histone marks that promoted their gene expression. These included genes PROX1, GABR2, KPTN, KCNA1, SNTB2. A number of these have properties that are associated with synaptic plasticity. Some of them are genes that also don't have much neuroscience literature behind them, so we're actually exploring these a bit more in the lab in terms of reproducing this with PCRs and determining the function of these genes. And same with some of the downregulated genes after exercise. What genes are going down and what new histone marks do they have? And so again, we found more than 5, but the ones that really stood out to us are listed here with new histone methylation Many of these have properties that promote inflammation, so actually are reduced after exercise, so some made sense. And some we're still investigating what their role may actually be in the mechanisms of exercise on hippocampal function. So we're really excited about this model that we've developed in the lab. But the whole goal of us developing this early life exercise model is to see if we can learn how these neurobiological mechanisms that are unique to early life exercise can be used in the setting of early life maladaptive experiences or early life neurological conditions that impact cognitive health. And so one of the models that we have brought this early life exercise paradigm to is a model that I worked on as a graduate student of limited bedding and nesting in rodents. And this is a model that promotes fragmented maternal care in the rodent parents. And you can see through the video here that the rodent mom is in and out of the nest in this limited bedding and nesting cage. She doesn't care for the pups consistently. And on the right-hand side of the screen, I just have some of the long-term outcomes of this model, including impairments in object location memory, impairments in dendritic structure, as well as volumetric changes in hippocampus after this chronic early life adversity invoked by this limited bedding and nesting model. So we are currently in the lab using our EMX1 new trap early life exercise mice, exposing them to this adversity model and exercise after the adversity experience, and seeing if we can reverse some of these long-term consequences of early adversity on cognitive function, on dendritic structure, hippocampal volume, and interrogating the mechanisms of that, which we can powerfully do with this new EMX1 new trap mouse. We're also taking this EMX1 new trap mouse to mouse models of acquired epilepsy. And so we have an early life model of status epilepticus. We know that in these models of status epilepticus during early life, we get a potentiation of newborn neurons in hippocampus. We also get a potentiation of newborn neurons in hippocampus with early life exercise. But it seems that these populations that seemingly do the same thing in hippocampus increase after these early life experiences have very different functions in hippocampus, especially as the hippocampus matures. So interrogating how exercise-induced neurogenesis and epilepsy-induced neurogenesis contribute to newborn neuron function and integration into hippocampal circuitry is another direction that the lab is going in. But with this model, we have shown so far that these canic acid-induced status epilepticus mice do have impairments in cognitive function when they're young adults. So we're also asking the question if we can reverse this with early exercise, and do the changes in neurogenesis and the mechanisms driving neurogenesis have anything to do with these cognitive outcomes after canic acid-induced status epilepticus. So these are just two examples of kind of the applications of our early exercise paradigm to pathophysiological models of cognitive disorders. And so to wrap up our take-home points, we have established that exercise elicits functional and structural changes in hippocampus to improve memory, and these mechanisms can be leveraged to inform long-term development and function of the hippocampus. And we went through these neurobiological mechanisms earlier on in the talk, including neurotrophic expression, enhanced synaptic plasticity, neurogenesis, as well as spine density and dendritic structure. The hippocampal formation, as I showed you, has a critical period postnatally for development, and there may be a sensitive period based on our early life exercise studies by which that exercise intervention can have a persistent impact on hippocampal function. And so emergent data from our lab is suggesting that early life exercise is influencing the hippocampus in unique ways to influence its long-term function, to influence its synaptic plasticity, as well as the epigenetic signatures that may underlie this early life exercise experience. And our goal is to apply this early exercise model to models of neurological and specifically cognitive dysfunction and the ways that we can model these within rodents in order to identify candidate mechanisms. So what are some of the open questions? What is the time course of exercise induced synaptic plasticity during development? And can we come up with exercise doses for treatment of neurodevelopmental disorders, and thinking also about exercise mimetics for those who can't? So with that, I would like to thank you all for your attention. And I don't think I have my thank you slide here, which is unfortunate, but really the most important slide is the acknowledgement slide, in which I highlight two graduate students in my lab, specifically Anthony Rouse and Nellie Nelson. These two students have worked so hard on developing our EMX1 neutrap mice. We also have a few technicians, Tim Yu and David Valientes, who contributed to this work. I'd also like to thank our funders, including the NINDS and the Robert Wood Johnson Foundation. So thank you so much.
Dr. Ivy, spectacular work. Congratulations. We have one question about whether you would predict that in a disorder of methylation, if you would anticipate that exercise wouldn't be helpful. So for instance, in Rett syndrome with MECP2.
Yeah, that's a really great question, and a really relevant question, because I believe Dr. Zogby has recently put out a paper in a very high-profile journal. It's either Nature or Science, I don't remember which, but it's one of those, in which she actually looks at behavioral and specifically motor interventions before the onset of Rett symptomatology in an MECP2 knockout mouse. And I believe it's a rotarod intervention that she uses. And what she finds in that study, and I would encourage you to look at it because it's a really interesting study. What she finds is with that behavioral intervention, the onset of the Rett syndrome kind of motor abnormalities seems to be delayed. So can there be a behavioral intervention that is done prior to the onset of symptoms to actually delay the onset of the disorder? I think we are starting to have evidence that that's the case. Now, whether that is working on, whether that's pre-symptomatic intervention is working on methylation that is, dictated by MECP2, or is invoking the activity of other enzymes involved in that methylation process, or histomodification processes. So changing the balance of methylation and acetylation on those particular genes, and specifically on the genes that are involved in memory, on synaptic plasticity and neuronal function, I think is something that is waiting to be explored. And I think that this type of work can allow us to do that.
Well, thank you so much, Dr. Ivy. I really appreciate this fantastic work.